I want to start showing, um, I can't move my slides. Oh, here. The burden of diseases in the world. And I just want you to see how the world from 1990 to 2017 has become more yellowish and less red. Even in Africa, that still holds the biggest burden and lagging behind, but the world is becoming more homogeneous towards the yellowish color. And the world was patchier than it is today, and we are going to converge eventually to the same color. And this holds, if you take, for example, Brazil, there is a nice public health paper in Lancet that shows the data. In Brazil, in 1990, Brazil was much patchier than today. And the reason is that the Northeast had a lot of diarrheal diseases that now have become a secondary or tertiary cause of leading causes of life lost. And ischemic heart disease is the leading cause. This is consistent with a much improvement in life expectancy in Brazil in these 26 years, from 68 to 75 years of life. If you take the regions and you see the top panel, you can see how in Brazil overall, the red line, which is communicable diseases, goes down. No change in the green line, which are injuries, but there is an increase in chronic non-communicable diseases. But the reason of that decrease in infections is really the states that had high infections earlier. So if we take the state of Maranhão, which is this a state that was blue and then become purple, you can see the decrease in infections. Sao Paulo, which has always been purple since 1990, there has no decrease evident in infections, but still chronic diseases are increasing. So even when we control infections, we still see an increase in chronic diseases. So humans, we speciated in Africa, and we were able to uh, inhabit all the rest of the planet, of the continents in the planet. And with these migrations, we diversified biologically, but also culturally. And today, urbanization is making us converge into a single lifestyle and again, homogenizing the world. So we are interested in studying the microbiome effects of urbanization. And the reason behind urbanization is really controlling infectious diseases and being able to live in big cities because we can clean the water by chlorination. We can use a lot of antibacterials in our canned food or in our houses to prevent degradation of the materials of the houses. We build our own environment in big cities. We have hospitals that have treatments that are antimicrobial, such as antibiotics. We use vaccines to protect us. So sanitation and medicine provides urban settings with the means to combat infectious diseases that used to kill our ancestors. So all these infectious diseases go down, but it's a trade. We are not getting necessarily better. We are changing them for diseases that have to do with the immune system, malfunction of the immune system, type 1 diabetes, autoimmune disease, asthma, celiac disease, food allergies, autism and Alzheimer are also in the rise. The explanation to these modern plagues are, as we call them, involve microbes. And there are two main hypotheses, both involve microbes. The first one is non-self microbes, is the environmental microbiota. The hygiene hypothesis is non-us. It states that reduced exposure to farm and animals increases childhood asthma and allergies. So we did a study funded by the Sloan Foundation to, to see how changes in the microbial and chemical world in which we live occur through an urban gradient. So we chose a gradient from the border between Peru and Ecuador, jungle communities, all the way east in the Amazon River, um, increasing urbanization to Manaus. And in that study, what we did was sample houses and their inhabitants, human and animals, across this gradient. In the gradient, we also saw how when you reach to Manaus, there are two Manaus, the middle class and the slums or poor people. So we had to duplicate in Manaus the sampling effort. So we published some of these data, and you can see from swabbing the walls of the houses, you can really identify the community to which the swab belongs by looking at their bacteria. The reason being increased human skin bacteria as you move towards urban and reduce soil bacteria. We enclose our houses more and more, and we separate them from the environment, an environment that in urban settings is much less natural, with less soil and less plants. We also study metabolites, and you can see in this principal coordinate analysis where each dot is a sample, you can see that from the red jungle community going to the intermediate Puerto Almendras and Iquitos to Manaus, there we move in the principal coordinate one to the left. There is a gradient of urbanization there. It doesn't matter the social class, low income and middle class in Manaus show the same composition of different metabolites. We observe an increase in the richness of metabolites to which we are exposed when we move from the jungle to urban. These metabolites are shown here in the left-hand side. We use a lot of products to clean, personal cleaning products, makeups, medicines. They all help to increase our diversity of our metabolic exposures. And we found on the right-hand side, there is a correlation between increasing our metabolic diversity and also increasing fungal exposure in our urban environments. So urbanization is changing our microbial and chemical exposures. The second hypothesis that has also to do with microbes is the microbiota, disappearing microbiota hypothesis, which states that it's lack of exposure to the normal indigenous human microbiota that disrupts the early pathways in life. And this, again, sanitation and medicine that can stop pathogens can also alter our good microbes. We have studied gradients of urbanization, and this is a composite slide showing the diversity of the fecal microbiota in very isolated Yanomami peoples from Venezuela, semi-transculturated Guahibo, or Malawi. So it doesn't matter the continent of the ethnic or the ethnic differences, they lose diversity, and the US here showing the lowest diversity in the fecal microbiota with westernization. This is a prevalence abundance curve just to show each line is one variant or OTU, one bacteria. And you can see how the lines reduce in prevalence and in abundance as you move from the green Yanomami to the yellow US. Here are some examples of disappearing bacteria, Helicobacters, the Sulfovibrio, Oxalobacter, and others. When What we did in this paper, which is the HATSA paper, what we did was to take all the sequences of fecal bacteria that had been published in and deposited in sequence data banks and order them in the principal coordinate one. And what happens is really astonishing, because what you see is a spontaneous ordering by positioning PC1 with the lifestyle. On the left, first, in the left, Venezuelan Yanomami that are very isolated, followed by other jungle savanna peoples in Africa or South America. Again, geographic location doesn't matter. Followed by rural peoples in South America and Africa, and then the urban cloud on the right, including Asia, Europe, and the Americas. With Ana Maldonado, a former student, we studied how Helicobacter pylori loss also leads to changing the microbiota in the stomach. These are Amerindians, H. pylori positive or negative, and each of these lines in colors are the different bacteria. With Daniela Vargas, we also have studied human vaginal microbiota, in part because indigenous peoples have a major cause of mortality, women, due to vaginal and cervical cancer. And the vagina is the primordial seeding source for future generations. So I'm very interested in understanding the differences in vaginal microbiota. Here in blue, you see the mestizo, and in red, you see Amerindians. But if you stratify the Amerindians by exposure, you see also the centromere, in this case, moving towards the urban. We currently, with Oscar Noya and Monica Contreras, and in my lab, we are now narrowing the exposure to urbanization to try to understand what is the effect without changing lifestyle. How do we change the microbiome? How do humans change the microbiota in the very incipient, which is basically, you can see the effects on the architectural and urban plan of the communities. You start seeing mixing materials and architectural design. But the most important one is medicine. Communities that have medical posts versus none. So these are our natural exposures we evolved to have. When we are born, the first time babies see microbes is during labor, and they get seeded with vaginal bacteria of their mothers. Then the mother puts the baby, exposes the babies to skin, to oral microbes. There was a lot of skin to skin contact until the baby has developed enough to explore. And the ways babies explore the world is very intensively, taking everything to the mouth, really recording the environment, which in the old days was very natural and full of soil and plants. In our urban societies, we impact every step of these sequential exposures. We use antibiotics in labor, we have C-sections, we use formula, we use technology, and we get dressed in ways that impair transmission, barriers of transmission of microbes. Our kids live in built environments that are extremely clean, except clean of natural bacteria from the soil. Babies sleep away from their mothers, and we raise them in really concrete jungles. We have studied differences of the neonatal microbiome first month, for example, in mothers that deliver with midwives, and Joanne Kombelik did the PhD with me, she's a nurse, a midwife, comparing home delivery versus hospital delivery. 10 mothers in each group, each mother giving birth with a midwife, all vaginal delivery, no antibiotics, all babies exclusively breastfed. To our surprise, we could distinguish the baby's microbiota during the first months. And I'm showing here on the right the discriminative taxa with the names. So babies that are born in the hospital, even with these very natural ways, had a lot higher representation of Enterobacteriaceae and lower Streptococcus, lower Bacteroides, and lower Bifidobacterium. If we do C-section, C-section involves antibiotics plus bypassing the vaginal exposure, plus early, uh, a little preterm, because C-sections, planned C-sections are done early. You can see in the principal coordinate analysis on the left, our study showing how the oral microbiota in green is different from the vaginal microbiota of the mother, and in blue, the skin microbiota of the mother. Now, we swap the babies in multiple body sites, and if they were born vaginally, they tend to cluster with vaginal microbiota. If they were born by C-section, they cluster with the skin microbiota. Their multiple body sites look like skin, and this is from the operating room exposure. We know that later, on the right-hand side, we see phylogenetic diversity, and we know that babies, both baby groups catch up, but they start differentiating again later in life after 8 months with lower diversity in the C-section. There is also evidence that C-section, like antibiotics Marty showed yesterday, accelerate growth. And same as antibiotics, there is epidemiological evidence in humans that C-sections, babies grow bigger, grow faster, and there is causal evidence in mice. C-section grow faster. So we started efforts of restoration some years ago, and we published the first pilot paper, restoring C-section born infants with vaginal fluids of their mothers. Recently, in December last year, the group of Willem de Vos published the fecal microbiota restoration. And we need clinical trials to see if this really means protection against diseases. There are many stressors in these industrialized societies that impact microbes. And we know from basic ecology that if you impact a normal ecosystem, the ecosystem is resilient and it recovers. But if you impact and before it recovers, you impact again, compounded impacts, there are extinctions. If you take a developing ecosystem that is not quite yet normal, mature, and you impact, there are extinctions. So our rationale was and is, if with industrialization, we are degrading the microbiota in ways that mean increasing the risk of modern diseases, the modern plagues, can we restore and revert this trend? And this, as we said in this perspective in science, will require collections and banking microbes from minimally disturbed traditional populations. So we need to collect, characterize in order to restore. And in this regard, the local collections in the world are extremely important. With this, I thank everybody for your attention, and my lab and the multiple collaborations that have made possible this work. Thank you.